Okay. Hello everyone, good evening. So let's just wait for another 1 or 2 minutes and then we can start. Okay. Yeah. Good evening, good evening. Tejaswini. Good evening, Virat. Creative shots. Good evening. I hope my voice is audible to each one of you and screen is clear. Please confirm me once. Voice is too low. Is it for everyone? Okay. Check like voice is low. Check now. Is it okay? Fine. It is fine now. How many of you are excited? Cover genetics in a day. Genetics in a hour. Will be like covering Mendelian genetics in mostly 1 hour, 1.5 hour. That's my target. Let's see like how it goes. And I won't be discussing anything much in detail because last at this last moment it's not even necessary. What are the required things? What are the things that are needed to be recalled? Let's discuss them in detail. Okay. Like I have made a PPT. I made a PDF of the entire things of Mendelian genetics that I'll be like presenting here, and based on that I'll be discussing using the same PDF. So if you want this PDF, do you want it to recall and to revise the things in last 1 week? How many of you want it? Molecular, started with Mendelian, you are already asking about molecular. Okay. So once the Mendelian genetics marathon is completed today, let's see like how you are responding, like how you people are responding. Based on that, we'll also come up with the molecular genetics. So how many of you want the PDF of this class? Irrespective of like whether you are a student of master course or not, how many of you need this PDF? Yes or no? Yeah, whole course, revising whole course, like it is possible now because I am a very much detailed guy. You ask me to just tell the things superficially of every concepts, I may not comfortably do it. That's why like this particular marathon series was also lacking, because it's explaining each and everything and completing it in one marathon, it's not possible. Again, like I always prefer to go for individual classes for different topics. That's the reason like we did not plan for the marathon, and we only had it in mind. This is also because of the student request that, sir, we want everything on a revision series. That's why we planned for the genetics marathon majorly, because completing course may not be possible entirely. That box is not visible. Breeding, okay. See, plant breeding, like we have come up with another alternative. Like for plant breeding, what we have done, so whoever has registered to our online mock test bundle, just the mock test bundle, even for them we have provided the detailed video discussion of the plant breeding topics. So if you are aware of it, so we have come up with the detailed discussions of plant breeding MCQs that were uploaded in the course. So the same, for the same set of questions, so we have discussed each and everything in detail. I hope like you are aware of like Abhishek sir has discussed in detail starting from the history till like breeding methods. Like in the upcoming few of the sessions, you will be covering all the leftover topics. Okay. Molecular is so complicated, it is also much needed. Tech also, breeding also, it completes the it covers the entire course. Okay, so let's see like how whatever is possible from our end. So we'll definitely try to make sure that we'll provide to you in this last time. But please make sure that put your efforts. Do not wait for just a ready-made food. So put your own efforts. Use your brain. Just try to revise and recall whatever you had studied till date, so that in the last one or two, like last 1 week, so you'll be able to make yourself most productive and revise whatever you have learned already. If you are getting any extra things, just try to get it, grab it. Okay. Not for seed science. Should I start the class or not? Yeah, just as I said, so today like we are starting with the Mendelian genetics. So based on the response, based on like how much time it is taking, so we'll plan for the molecular genetics, population genetics, and also like first part of genetics, which is the general topics, or also like with the cytogenetics that we can plan. Okay. Okay, good evening everyone. So let's start today's class. So let me introduce myself. I'm Hariprasad, a PhD student from Indian Agriculture Research Institute from the Department of Seed Science, not the genetics, but still I'm here to just teach you the topics of genetics. And why am I here? Like, because I'm interested in the subject of genetics. And if you know, like I teach genetics and also the seed science in the online master course of plant sciences in our platform AgriAddict. And this is my like a fourth year of teaching in the same field of genetics and seed science. Fine. That's a brief about myself. Let's begin. Okay, starting with let's start with the terminologies. So the important terminologies, we are going to start with the genetics. What is genetics? If you know genetics, who gave this term, and just post it in the comment section. It was by Bateson. And the allele is also a simplified form of allelomorph. So don't get confused with these two terms. Both are same. And allele, what is this allele? It is an alternative form of gene. If you know about a gene, if A is a gene, it has the alternative forms which can be noted as capital A and small a, which are called as alleles. Okay, and the genetics, I told you about the term, but what about the definition? You know, it is a study of what? Study of heredity and the variation. Okay, that's done. And the genetics term was derived from which word? It is from the Latin or Greek? Someone comment? And what does it mean? So hide the notification. Close the exam. Okay, it's from the Greek word. Okay, so what does it mean? Geneticos, right? Okay, so next, the genotype and the phenotype. What is genotype? Genotype is a genetic constitution, we can say. So what is genetic constitution? If I have a gene of A, so whatever we represent in the form of capital A, capital A, capital A, small a, small a, small a, this is called as genotypic representation. Next, what is phenotype? The phenotype is same here. This, if it is like brown color, then the other one will be brown, and next it can be white. So we are here differentiating or denoting it in terms of the physical appearance, so which is for the phenotype. Okay, next, so F1 phyla allele, F1, F2, F3, you might have heard about this. The breeding methods, what are these? These are derived from the Greek word, so sorry, from the Latin word, which is like phyla, so which means the son or the daughter. Like, who gave this term? This was also coined by the Bateson. Okay, next, who coined the term gene? Gene and the genotype and phenotype. So these two terms also, who coined these two terms, genotype and phenotype? If you remember, so these two terms were again coined by, what is the name? It's a William Johnson or Wilhelm Johnson. These spellings are also very much important, so please make sure to remember this. Wilhelm Johnson. What is the spelling of Johnson? So this is it. Okay, remember this as it is. And the gene, the basic unit of inheritance. This was a very basic definition. But what is the modern definition of a gene? Anyone? What is the modern definition of a gene based on the current understanding? So which was came after which experiment? The latest understanding of a gene, or what we call as modern definition of a gene. It is with the experiment of changing concept of a gene, Benzer experiment, right? That we'll discuss in the later parts. Okay. Next, the term homozygous and heterozygous. This is also very much simple. I won't be reading any of the definitions. Just see it here. See, homozygous is something which has identical alleles. So this is the homozygous, and heterozygous, it is having the different alleles of a same gene. So that is what the and hetero means. Hetero is different, homo is same. So this is all about the definitions. Next thing, what about Mendel? So everyone who is reading for these exams of plant sciences, you should be very clear about the Mendel's very born. So regarding his history, the schooling, and which of the experimental materials that he have used for the study. First thing, Mendel, birth date 20 July 1822, and where he was born. So the current place where he was born, it is like earlier it was Austria, now it is what? Czechoslovakia, right? And the place name is now Bruno. Brunn was earlier, now it is called as Bruno. Fine. So who was he? He was a Augustinian monk, that we already know. But what else was a major contribution of the Mendel? First name, full name. What is the full name of Mendel? It is Gregor. Again, what is the spelling? Johann Mendel. Right. So he was an Augustinian monk. And next, what he has done? He has given the the two important laws of genetics. Actually, it is three, but only two are considered as a universal laws. If I have to read all three, it is law of dominance, law of segregation, and law of independent assortment. Okay, this is fine. So what are the different experimental materials he has used for his experiments? On which of the organisms he has done the experiments? Can anyone tell me? Yeah, 1884, good. The death. Okay, fine. So the experimental material which was asked in one of the previous year exam, one you all know, it is about the P. That's fine. But what about the other experimental material which you should be aware of? Yes, mice or rat, or next, what else? Rajma, physalis, ericium, okay, awkward, and okay, mice, rajma, okay. See, I think like many of you have seen the honeybees in the books also. Might be in some books, but not at all mentioned in any of the standard literature that was mentioned. So there is a lot of confusion regarding the honeybees. But if it is asked in the exams, they won't confuse you with the terms which are not actually proved experimentally. So you need not to worry about it. But make sure these are the list of the experimental materials which the Mendel has used. Okay, so he has given the law of segregation and law of independent assortment, which are considered as two universal laws, right? So these are laws of genetics. We are famously called as Mendel's two laws. And what are these laws suggest? Let's discuss in detail in the upcoming slides. Before that, so talking about the timeline, you know that it was in which year this laws of Mendel was proposed. So it was in 1855 or 56. So in which year the Mendel's laws were proposed? Okay, is it in the 1865 or it is in the 1866? So in the 1866, what important event has occurred? So it was published in which journal? Published in what? Published in Annual Proceedings of Natural History, Proceedings of Natural History Society, right? So if you remember, but before it was published. So what is the importance of 1865? It was on two different or two important dates, Feb 8 and March 8. It was presented in the annual meetings of the same society. But in the next year, in the following year, this was again published. So this was the importance of it. But as you know, like even though it was published, it's not like that the work of Mendel was not published, but it was published, but not got a widespread acceptance to the work he has done. Okay. Next, this was published in which language? Someone has rightly said it. It was published in German language. Okay, remember this. Okay, this is very much important. Whatever Mendel's has given the laws, he was since a German, like it was proposed in the German language. But since it got a widespread acceptance only after 1900. So what important event has occurred in 1900? The same work was independently rediscovered by three different scientists: Hugo de Carl Correns, and Schermark, right? And Carl Correns was from Hugo de was from Holland, Carl Correns was from Germany, and the Schermark was from Austria. So this was rediscovered independently by these three different scientists, and soon after its rediscovery there was one more important event that occurred by Bateson. What is it? It was translation. What translation? Translation of the Mendel's paper from German to English, where it got widespread because of this translation, since the English was accepted everywhere or most of it. Okay, please erase the close the Telegram, close the app. Yeah, or just remove the notification from there. Okay. So this is about the translation, this is about the Bateson, this is about the rediscovery. It was in 1900 by these three different scientists. Also, one more important thing that you have to know, it is independently. And also along with this, this is one more Spillman, William Spillman. So even this scientist name is also pronounced for the rediscovery or the credit for the rediscovery of the Mendel laws, but as it is not published or the non or the direct credit is given. Okay, so this is about the total history of the Mendel, like how it born, and he died in 1884, and if you know, it is after 16 years of his death everything got noticed. His complete work was noticed and it was credited, and currently, if you know, is called as father of genetics for his remarkable work he has done for giving the two different laws of genetics, fundamental laws which tells about the heredity and the variation, two laws of Mendel. Okay, fine. So same thing have been posted here. So one thing to note, he was called as brilliant. Why he was called as brilliant? Because first thing, I told you in so many times in the class that he has analyzed the previous work, he has done the perfect study, and he has made the careful execution of the experiments where he has kept all the data related to the experiment, plus whatever numbers that he was getting, since the numbers were huge, he applied mathematics to biology and converted it into ratios, and then he have made the insightful interpretation of whatever results that he has taken, and he has studied a single trait, the monogenic, the single trait at a time, did not concentrate on multiple traits at a time. So one thing I missed. So how many traits did the Mendel studied here? Total, he studied the 7 different traits. So what is important for your exams? You have to remember all the 7 traits which are there and the corresponding, the currently new chromosome on which they are located. And if you know, like out of the 7, the 4 genes have been cloned successfully, rest are still pending. Okay, so what are the 7 different traits that are listed or studied by the Mendel? I'll lead to you, so just make sure to recall it and read it once. So there might be a lot of confusion. I'll try to clarify some of the confusion in this video. Rest, if you have any doubt, you can comment on this video. I'll be happy to assist after completing this live series. Okay, next, so this is about the why he was brilliant. You can get these questions in terms of like other assertion reasoning or in the statement wise questions. So make sure that you try to remember this and try to understand this, because if it is asked, you have to answer it. Okay, so next, the Mendel, he was also considered extremely lucky. I have mentioned only 3 important points, like several points can be also added based on their personal interpretation. So here, first thing, it was concluded in 1863. Whole experiment was concluded in 1863. In the following year, the pea weevil destroyed almost all the pea plants in the following year. Next, the 7 traits which he has like studied or which he selected for the studies were monogenic with the complete dominance. Okay, but out of the 7 traits, one had pleiotropic effect. Which is it? Which of the 7 traits of the Mendel had a pleiotropic effect? Okay, good. The one which is seed coat color, or the pleiotropic effect with the flower color. A single gene is providing the 2 different types of phenotypes, responsible for 2 different phenotypes. One is 2 different traits, so which is like seed coat color, and other one is the flower color, or whatever you can say, like you can say it as 2 different characters. One is seed coat color and the other one is the flower color. Okay, so this is about the 7 traits. And the next thing, it did not encounter linkage. So what is linkage? So I'll discuss in the upcoming slides. Is it clear to everyone? Okay. Next, YP, the scientific name, Pisum sativum, and also you have to know the chromosome number. So what is the chromosome? Diploid chromosome number of Pisum sativum. So why Pisum sativum? Three important points again. So this can be easily grown, and the flower contains both male and female parts. It is hermaphrodite, and it is like purely self-pollinated. That's the reason why he has choose and why it was easy for him to conclude the experiments with the P, Pisum sativum. Next, how many varieties actually the Mendel worked on? He actually collected 34, in which he has selected 22 varieties, which like he has made his study. And the paper which was published, it was with the name experiments on plant hybrids. So it can be mentioned as plant hybridization in some of the books. So do not worry about whether it is hybrids or the hybridization, because in both the things are mentioned and something as incorrectly incorporated. So what is the actual definition or actual title? Title is like experiment on plant hybrids. So this is from the German translation. Okay, so whatever German title was there, when the exact translation is made, so it is experiment on the plant hybrids. Next, the Mendel's work was rediscovered, as I mentioned. It was by the three important scientists, Hugo de Vares and Karl Correns and Erich von Scherman from the Austria. So these names from Germany, from Austria and the Holland. So this is also important. Please try to remember it. Okay, fine. Next, I told you about the spill map. If you know about it, remember. If you do not know about it, you can entirely skip it, not an issue. Next, the 7 traits which I was talking about, the seed shape, seed color and the flower color. Do not get confused like why there is the missing of seed coat color. You should be knowing it here. It is seed coat color and for the flower color both. And the flower position, plant height, pod shape and the pod color here. And what else is important along with this to know about this dominant trait and the recessive trait? Round and wrinkled for seed shape, the seed color it is yellow and green, and for flower color it is purple or gray and white, and the flower position it is axial and terminal, plus the plant height it is tall and short, the pod shape inflated and constricted, and pod color it is again green and yellow. Okay, try to remember this traits of both dominant and the recessive. Okay, so now the pleiotropic effect is also mentioned, so I have already done that. So next, let's talk about the law of segregation or law of purity of gametes. So what according to this, I'll try to explain this in a simple words. See, if there is a genotypic constitution in a plant which is capital A and small a, so you know like while going for the next generation, it has to form the gametes. So while forming the gametes, if you know like the gametes will be in the haploid condition, not in the diploid condition. So while forming the gametes, these two alleles do not mix with each other. They will be separated from each other in a equal proportion, and then it will go to the next generation. It will go to the next generation without mixing with each other. So this is what the law of segregation explains in a simple words. Okay, next, quick evidence for the equal frequency of the gametes. So this is the one thing that you should be knowing. So I told if there is a capital A small a in an individual, for example, it is giving some color. Let's talk about the color here, red color. So I told it will form the gametes, but how will we know that where the gametes which are formed? Let's assume that the total 100 gametes are produced from this individual. So whatever gametes that are formed, how will I know that it will be equal in proportion? 50 will be capital A and 50 will be small a. So how will we know it? Yes. So this is because of by knowing the indirect evidence, which is the test cross. See, whenever we do the test cross, what is test cross? Crossing the heterozygous with the recessive parent. So once we are doing it, if this is produced in the equal proportion, only then the result will be 1:1. If this ratio is not in the, or this gametes which are produced, it is not in the equal proportion, so then we won't be getting the next generation in 1:1. So what I mean to say, see if you cross this, I'll show you. So if you cross this, you know, like these are the possibilities, either capital A and small a, and here the gametes will be only capital A, only small a. So if you do it, it is capital A small a and small a, which is exactly in 1:1 ratio. This is only possible if there is an equal frequency of the gametes, but this is actually a indirect evidence. So what is the direct evidence for this fact that gametes will be produced in the equal proportion? Okay, waxy gene in the maize. I talked about a very important one which you can remember. This is segregation of the spore for the neurospora, which actually showed a direct evidence regarding there is a equal formation, equal frequency of the gametes have been formed. You can remember the examples. So this is test cross. You have to know its practical implication, that it is used to know that whether the, and oh, what else other than the test cross? What it is? What is the application of the test cross other than knowing the equal frequency of the gametes? What do you think? Other than knowing the equal frequency of the gametes, what else is the application of test cross? Knowing the genotype. You remember what I have said in the class, knowing the genotype of the parent, right? So remember this point too. Okay. So what are the exception of Mendel's law? See, whenever we talk about the exceptions, there are like lot of things that are present in the different books, right? But I want to, like, let me make it very clear and very simple here. The exceptions you can take, one is paramutation for the first law, and second one is non-disjunction. Third one is meiotic drive. I understand, like even like in my notes, like I have given a lot of other exceptions too, but from all of it, so you can remember these three. These three are the most important one. So what is paramutation? If you remember, there are two different alleles: paramutable allele and the another allele which is undergoing the mutation. So here, if there is a capital A and small a, so this has the ability to mutate the other allele. So one allele has having a effect of mutating the other allele. That's what the paramutation is. So since they are the alleles of a single gene A, so this will be an exception for the law of segregation. Why? Because the law of segregation is talking about how many genes. So the law is for how many genes? Is it for the two genes or a single gene? Whether the law of segregation is explained for a single gene or the two genes clubbed together. Yes, quick. Yes, it is for single gene. So that's how you can directly relate the paramutation concept here. What is non-disjunction? If you know the concept of non-disjunction, which I explained clearly, that the alleles of a gene during the meiosis or during the cell division, it won't separate, or it is more related to the chromosomes. Where there is non-disjunction, the two chromosomes will go to the same pole without separating from each other. That's what the second exception is. Third one, what is meiotic drive? Can anyone explain what is meiotic drive? Meiotic drive is something where if there are two alleles, let me explain in the very simple words. If there are two alleles, one allele will suppress the other allele in passing on to the next generation during the gametic stage. So while passing to the next generation, this will suppress, this will not allow the other gene. It's not always dominant, it is like random, okay? But there is a phenomenon called as meiotic drive where there won't be like equal passage of both the alleles to the next generation. That is what the meiotic drive means, okay? So these are the direct exceptions of the Mendel's first law. Next thing, the second important thing is like independent assortment. Okay, yeah, meiotic drive is also called as segregation distortion. See, when we are talking about independent assortment, like I told you, I did not talk about the linkage. So why? Tell me whether the linkage is an exception for the first law or not. Anyone? Linkage, whether it is an exception for the first law or not. Yes, why no? Because linkage is a phenomena which comes when there are two genes. If I have taken two different genes, if there is a chromosome and I am talking about minimum of 2 genes, then the phenomena of linkage comes. Otherwise, linkage is not having any importance here. Okay. The next thing, in the independent assortment, what we are talking, like we are talking, right, that if this is a gamete which is formed and this is a gamete from the one parent, parent 1 and parent 2, they are separating from each other and they are going into the next generation. There is an independent assortment, so because of which we see the different combinations of the progeny in the next generation, where the segregation of 2 or more characters in a same hybrid is independent to each other, because here they have taken off round and yellow, two different traits or characters. So here these two characters are not mixing, they are being independent to each other and they are passing to the next generation. That's what the independent assortment means. The second law, what is the exception for the second law? So these are the exception. So one thing, the important thing in this is knowing about the linkage, the epistasis. So this phenocopy. So these three things are important. Rest you can remember it as it is. Okay. Next, very important formulas. See, this is very much important. Please try to note it down as it is, because you can expect the questions directly in the exams and there is nothing much to apply your mind, so you can remember this directly. Okay. So the second, starting with the different kind of gametes. So total number of gametes, we just directly do it as 2 power n. So this is, you might be knowing, and for the phenotype is 2 power n, genotype is 3 power n, and total number of the F2 combinations that are possible. The perfect, you can also have the word called as perfect here in the exam. If it is, they are asking about the perfect F2 combination, like number of individuals, it is 4 power n. And in the F2 individuals, like how many are homozygous, it is directly like 2 power n. If they ask you about in F2, like how many are actually heterozygous, then you have to minus 2 n, 2 to the power of n from the 3 to power n. Okay. So now what is n here? I did not talk about it. Can anyone tell me like what is n here? What is n in this case? Heterozygous genes. Number of genes or number of heterozygous genes. Okay, good. So next, so if we want to calculate for the quantitative trait, so this is all for the qualitative, right? If you want to do it for quantitative trait, then it is like 2 power 2n plus 1, okay, directly. And the number of phenotypic classes, it is again 2 power, this is the same. And the last three, last two, it is related to the chromosomal trisomy, right? What is trisomy? If you remember, it is having the extra chromosomal set. I have, although like I won't be discussing anything about cytogenetics in this video, but still, since the formulas are at one place, like I have tried to include these two. But here you can see that there is a missing of the tertiary trisomics. So what is a formula for calculating the number of tertiary trisomics? First thing, what is trisomics? Trisomics is nothing but having the extra chromosomal set where it will be 3 instead of 2, right? That is what trisomy means. So in that we have three different categories: primary, secondary and tertiary. So I won't be discussing this here now, but let at least tell me like why I have not mentioned the number of tertiary trisomics here. It can be with the any number, okay, or it can be many or infinity. So that's why no formula for that, as simple as it. Okay. Next, application of the Mendel's principle. So what are the applications of Mendel principle? How to know the outcome of a cross? First thing, like the application of the Mendel's principle can be applied to know the outcome of a cross. So if in genetics, if I have to talk about, if I cross capital A small a with capital A small a, so what is the outcome? How can I know it? So this can be known by using the three different methods. In a very simple words, the first method, it is like Punnett square method, if you know about it, and the second method, it is called as fork line method, and the third method, which is the quickest method of knowing all of these crosses, it is log probability method. And this is more suitable if you have one or like just two genes. It is like two or more genes, for two or more genes, for this is for any number of genes. Okay, just try to remember all these three and how we do it. Like if I have to talk about Punnett square, this is how we do it, right? So this is like capital A small a So this is capital A small a, and again above is capital A small a, and we just try to cross it and we get the answer. Where? So this is 2, 1 is to 2 is to 1 in case of genotype. If we talk about the phenotype, then it is 3 is to 1. This is about the Punnett square. Four-line method. So if I have to do same thing, this is like, you know it, like it is A and it is capital A small a, and it is capital A small a, and it is small a small a. Okay. So this is how we can use the two methods. What about the probability method? So if you are aware of probability method, so in case of probability method, so we do it in a fraction. So we just try to multiply the fraction, and it is most useful when there are more number of genes that are need to be done. So please remember, these type of questions will definitely come in the exam. So you should be very much aware of this fact, like how we are going to do it. For example, so if there is capital A, capital A small b, capital C small c, which is crossed with capital A small a, capital B small b, and small c, small c, small c. So now in this outcome, if they are asking about the specific combination, what will be the frequency of capital A, capital B small b, and capital E small c. So how you are going to do it? So this here in this combination, you have to know this combination is 1, because when these two are crossed, you will get 1 here, and it is like 1 by 2, and this is 1 by 2, and this is same, like it's 1 the probability, and this is like half, and it is like 1. So this is how you can do the probability method here. Next, testing of genetic hypothesis. We come to the next part. Here in the testing of genetic hypothesis, what is it exactly? See, from the Mendelian cross, you know what is the ratio. I did not talk about the ratio. Okay. So first law, according to the law of segregation, we know that it is like 3 is to 1 for the phenotype and 1 is to 2 is to 1 for the genotype. Again, in the second law, what is the ratios? The genotypic and phenotypic ratios in the F2, it is like 9 is to 6 is to 1, right? So now 9 is to 6 is to 1 ratio, like if we get it whenever we cross the parents, and then we come into the conclusion in the F2 that it is actually 9 to 6 is to 1, then we can assuming that okay, certain these plants, these traits are actually following the Mendel's laws. But every time do we get exact numbers, exact whole numbers without any deviation from it? Because here you can see that this is the Mendel's dihybrid cross. Okay, what was expected according to the ratio of 9 is to 3 is to 3 is to 1. Sorry, I mentioned it is 6. Okay, 9 is to 3 is to 3 is to 1. So according to the Mendel's dihybrid ratio, like the expected ratio is this and the expected numbers are here. But there is some deviation for the numbers, although like deviation is not much, but there is a deviation. Similarly, when we go for the Ugo-Divares dihybrid cross, so this should be again, see how much difference there is from the observed and the expected. So now if such the case that there is a difference between the observed and the expected, then how we are going to finalize that okay, this population or these traits are actually in accordance with the Mendel's principle. So how we are going to do it? So that's what the testing of genetic hypothesis means. So for here in this particular case, tell me the test, by using which test we are going to prove it. So yes, this is by the chi-square test. So what is chi-square test? Here we get the two different columns, which is expected, observed, and the expected, and the formula says the chi-square, it is the sigma of the observed minus expected whole square divided by expected. If we do it, we get some value here, and the value we have got, we have to look into the table of probability. There is some table. So in that table, it shows the significance level. So the table value for the different degrees of freedom. What is degrees of freedom? Again, that depends on the genes, number of traits we are taking. If the number of traits are 2, what is the degrees of freedom? Or the formula for degrees of freedom, it is like n minus 1. So now if it is the formula is n minus 1, where n is like number of traits we are taking, then what is the degrees of freedom for that? It is 1. So we have to look in the table value for degrees of freedom 1, what is the value, and the value which is present in the table, this value which we have obtained from the chi-square test, that should be less than this value. Only then we can assume that following interpretation is correct and the experiment that we have conducted is in accordance with the Mendel's laws. Otherwise, if the chi-square value is more than the critical value on the table, then in this case the above experiment is wrong or something is wrong, where this is not actually following the Mendel's principle. So that is how we can conclude. So for your exam, so you have to remember this formula and how to know whether there is significance difference between the expected and the observed or not. See, if the chi-square value, I'll repeat again, do not get confused. See, let's assume like we come up with some chi-square value. Let's assume it is like 0.5. Okay, and the critical value for this is around 0.6. So if this is the case, then here you can see the chi-square value is less than the critical value, right? So if such the case, then there is no significance difference. No significance difference means no significance difference between the observed and the expected values or the expected numbers. If there is no significance difference, that means the following calculations holds good or the following experiment holds good. So it is a fact that it follows the Mendel's principles. But if I say that here it is like 0.7, where the chi-square value is more than the critical value, that means the whole experiment is wrong or something is wrong in that, and that's the reason it is not following the Mendel's principle, and that's what we conclude, and that's how we conclude it. Okay, fine. So next thing, the testing of genetic hypothesis, same thing I have tried to simplify in a single word. See, don't expect that like I'll be able to explain each and everything in detail. That's not necessary for now. Like you have at least 3 to 4 days. If you just try to focus on one subject and try to break it and try to read it in detail, so you won't be at the end of the day you won't be able to remember anything. So that's why I'm trying to highlight the things. At least these are the minimum things that you have to remember, and that will definitely help in your exams. You will definitely say this after completing your exam. Okay, and who gave this chi-square test? Yes, good. Someone has Purna has answered it, I guess. Yes, Purna. Fine. This is by Carl Pearson, the year 1900. Okay. See, along with this test, one is chi-square test, but along with the chi-square test, also know about F test, T test. These are also important. So where you can do it? So you can do it on our YouTube channel. So there are the stats lectures which are uploaded. So they are just of 6. You can just finish it in 2 to 3 hours. Try to watch all the stat videos. So any questions that are coming in the exams, you can answer it. And this chi-square test, what is the other name of this? Goodness of fit. Also know this. Goodness of fit test. Okay. So the procedure and everything you can leave it, but remember this critical value which I talked about. For the field experiments that are conducted, we assume the critical value. We see the table value for the 5%, and for the lab experiments which are mostly accurate, so we get the value critical value of 1%. What is this critical value? Why we are doing it? All these things are not needed now. You can skip it for now. But once you go into MSc, like if you are pursuing your MSc from genetics and the plant breeding, and for all other subjects, this statistical analysis will be most important, because at the end of the day, once you complete your research, so the statistical analysis will be the one which you will be doing. So once you come into the MSc, please make sure to have a clear-cut understanding on the statistical procedures. The next is gene symbols. See, for gene symbols, why it is used? For the clear descriptions, so that there will be no doubt while understanding a concept or explaining the concept. So this is why like gene symbols are very much important. And the first monohybrid, first we'll talk about like how the gene symbols are involved, like introduced in the monohybrid. For monohybrid, so we have like, for example, there is one gene and with the 2 different alleles. So how we are going to do it? Name it. See, whatever might be the trait, like for example, it is round and wrinkled. So how we are giving the gene symbols? See, we simply mention it as capital R and small r, where capital R is red, round, and small r is wrinkled. So directly these symbols, the letters are directly taken from the trait, which is the dominant trait. Next, if it is for mutant, mutant is dominant here, like now for the notch wing, where like n is taken, capital N and the small n. And if the mutant is recessive, for the vermilion, that doesn't matter whether the mutant is like dominant or recessive. The gene symbols, the one way, there is no exact correct way. All of these are correct, but they have been changed from time to time, and for the different things, the different gene symbols have been used. So one example was directly using the dominant and the recessive, where the dominant trait will be used in the alphabet. But this is the one thing. The second, mutant can be also used. For example, for the notch wing, as I said you, like it has a gene symbol of N. For the vermilion, even it is recessive, it has a V. So this is how like we give the gene symbols. And for the encoded protein, so like if some gene is encoding a protein, then the gene symbol can be derived from the protein which is encoding. For example, P34 and P35, where 34 and 35 is the mass of that particular protein. Next, we also denote it as a superscript. Like if you know what is plus, plus always denotes the wild type and minus denotes the mutant. So this is the another very easy way of representing or giving the gene symbols. But for the multiple alleles, when there are multiple alleles for a single gene, for example, fur color in rabbit. So how we are going to do it? See, when it is wild type, either we'll go with plus or it is C plus. And when it is mutants, so like albino, the second one, this is just small c. If it is Himalayan color, CH, and it is chinchilla, it is CCH. And one thing, please make sure to know that Himalayan here, it is denoted as CH. Okay. And the chinchilla, it is denoted as C superscript CH. Know the difference here, because here for this particular trait, there is a dominance relationship that you also should know. Since the dominance relationship is also important, where the C, which is the wild type, is most dominant than all other types. I'll let you know like what are the other types in the upcoming slides when we talk about the multiple alleles. That's why this gene symbols becomes very much important. Next, the molecular basis of dominance. Here, this part we'll skip it. We'll not go through this in detail. Next, directly we'll talk about the extension. The molecular basis of dominance. Like I'll give you some introduction. So, what is it exactly? We know that when there is a trait, for example, when there is a gene, let's take it as R this time. R refers to the color, seed color or flower color, anything. Let's take it as seed color, capital R and small r. So these are the two different classes of the particular character. Now, this in the homozygous condition, it's very easy to understand it is red. It's in the homozygous smaller, it is easy to understand it is white. Here in capital R smaller, yes, we know that it is red, but why it is red? How it is red? What exactly is the molecular mechanisms of making this as a red? That is explained in the molecular basis of dominance. Okay, that's it. Now, extension of Mendelism. So these are the terms or the phenomena which have been not actually directly explained from the Mendel laws. Okay, the first one is the incomplete dominance or the partial dominance. Second one is codominance. The third one is O-dominance, multiple alleles, pleiotropy, and lethal alleles. That's we'll discuss one by one. First one, incomplete dominance. Let's take a one proper example and discuss all of this. Let's take R is a gene which has a two different alleles, capital R, small r. Okay. For example, like incomplete dominance means whenever you have three different classes, so in these three different classes, the heterozygote should have the dominant phenotype, right? If it is completely dominant, but since it is partial or incomplete dominant, so the phenotype that we are going to observe here, it won't be neither from the dominant nor from the recessive. It will be of a intermediate type. For example, if it is red here and it is white here, it can go for a pink, where the color pigment, since it is talking about the color pigment will not be able to completely mask the other one and express itself. So that's why we are giving the pink color. So in this case, even here, because of the incomplete dominance, we are getting the three different classes, phenotypic classes. So why I'm mentioning this? So in the exams, if they are asking for a complete dominant relationship, what are the different phenotypic classes we are going to get in the F2 for a single gene? For complete dominant, if it is completely dominant. What if it is incomplete dominant? Different phenotype in F2. What you will answer? Tell me the first one. For the complete dominant, what will be the different types of phenotypic classes? And for the incomplete dominant, what will be the number of phenotypic classes? We are obviously talking about the qualitative trait. Do not get confused. So here it is simply the 2, and the second case it is 3. As you can clearly see here, one category is, one class is red, second class is pink, and third class is white. Okay, so this is about the incomplete. Codominance, even in the codominance, how many different classes we are going to get? Yes, talk about the classes. 3. But there is a significant difference between the mechanism or the understanding of the partial dominance and the codominance. Here in this case, this capital R, small r, what it will do, both the alleles will actually contribute to the trait. Both the alleles will have a equal contribution. That's what is in case of codominance. But in case of incomplete dominance, if you remember, it's not about the equal contribution, it is about the incomplete masking or the incomplete masking, or it is the incomplete expression, we can say, because of which the color is not completely red, it is pink intermediate. But in case of codominance, both are contributing to the trait. That's why this particular trait of the middle class will change. So one good example to know about the codominance, what is it? One good example for the incomplete dominance. Blood group, good. So one good example for the codominance is a blood group. And what about the incomplete dominance? Mirabilis jalapa. Okay. So which is actually a red, pink and white color. Okay. So this is same. And for the codominance, it is blood group, ABO blood group. I'll show you like how exactly it is in the upcoming slides. So next comes the O dominance, where the similarly, like if it is capital R, capital R, capital R, small r and small r, small r. So this will be like O dominant to that of the homozygous one, where again how many different phenotypic classes you will observe? You will observe 1, 2 and 3. So this is in case of codominance. Next, multiple allelism. See, do not get confused about the multiple alleles concept. See, multiple alleles, what we are talking about, whenever we talk about the alleles, they are alleles of a same gene. So again, we are talking about a single gene, a gene, but generally or a standard representation is like we have seen that it is having two different alleles. It is capital A and small a. But for certain traits, there will be, for certain characters, there will be a involvement of multiple alleles. That means, for example, there is, we take another gene B, then it can have 3 different types like B plus, B minus or small b. Any number, any kind of notation we can give, but what it simply means, it means it has a 3 different traits for a single gene. That's what it means. Is it compulsory that it has to be just 3 or more than 2? More than 2 will be multiple alleles, or the 3 is multiple alleles? Having 3 different alleles is multiple alleles. What is the case, or what is your understanding? Yes, it's just the more than 2. What is the very good general example for this? It is the fur color which I told you in the rabbits. Okay, that you can remember. And also please make sure to remember all the examples. Do not try to search the examples, try to remember the examples from the BDC book. So that's more than sufficient. You need not to go anywhere and search for anything else. Just try to remember the examples from the BDC book. Next, we have a pleiotropy. Pleiotropy is another very good example. I'll tell you the phenomena, and then next is lethal alleles. I'll go to the next slide and explain it. So before that, it was in 1902 which I told you about the translation from Mendel's paper by the William Bateson. Here it is not Wilhelm, here it is William. Remember, do not get confused with the terms. It is William for Bateson, it is William for Johnson. Okay, and this was with the title, so title which the Bateson given. Remember the title also, the Mendelism, the principles of dominance, segregation and the independent assortment. So this was a title that was given in the translated paper. But what was the title of the original paper from the translation? Experiments in the plant hybrids. Okay, this thing you have to remember. And again, there was a, like, after the one paper that he was translated, he has also done the experiments based on the Mendel's laws, and he has followingly published the other papers too. Okay, where it was like just to support the Mendel's work and to propose the supporting evidence for his work. So that's what most of the scientists have done after 1900 or 1900. Next, the incomplete is done, codominance is done, O-dominance is done, multiple allelism is also done. Here I told you to remember about the dominance relationship. So here the C plus is dominant over the rest, and this one, whether it is Himalayan or Chinchilla, what is it? The second one, whether it is Himalayan or Chinchilla. I told you in this one. What is it? Yes, it's Chinchilla, and CH is Himalayan, and small c, it is albino. Albino is a color or it is a colorless? Why do we use the term albino? Albino or the white, the very common terms that we use in genetics for the different traits. Albino or we use white. This means that the color is actually white, the color is actually albino, or it is for the colorless. Yes, it is, because it doesn't produce any color. So that's why it is either albino or colorless. See, you cannot see any color here. The original color or the wild type color is actually the brown color, but here there is no production of color. That's why you do not see anything here. That's why it is either denoted as albino or the white. So here I was talking about the pleiotropy. In the pleiotropy, the single gene, if I am talking about a single gene with the multiple traits or the characters, it will have the same gene. For example, if I talk about same gene, it can have an effect on, let's take a same example, seed coat color. It can have an effect on the flower color. Also, it can have an effect on the stem length. So this is a hypothetical example I'm talking about, but these two are the one which is already there. We have discussed. So this is what the pleiotropy means. Next one, lethal gene action. So this is important. What is this lethal gene action? What is lethal genes? Lethal genes are those which cause the death of the organisms if it is present in that organisms. That's what the lethal genes means. So again, the lethal genes can be grouped into 5 different categories. One is lethal, semi-lethal, subvital, vital, and supervital. Lethal is all 100% of the organisms die if it is present. Semi-lethal is like more than 90% of the organisms dies, and it is less than 90% if it is subvital. Vital, like 100% will survive. There is no death. And supervital, it is example, remember the Superman, where the functions of the traits will be like extraordinary, like extra than normal, not ordinary, it will be extraordinary. That's what the supervital means. Now in this case, in the lethal genes, if they are completely lethal, they are divided into 2 different types again. So what are the 2 different types? I'll tell you. So you might have heard about 2 important terms that you have seen during the experiments. One is called as, so here in the lethal genes, you might have heard about 2 different terms. One is recessive lethal, the other one is dominant lethal. Can you tell me the difference between the dominance lethal and the recessive lethal? So what is dominant lethal and what is recessive lethal? If we are talking about a gene called as Y. So now I'll tell you the genotype here. Here in this case, capital Y, capital Y, capital Y, small y, and small y, small y. If this small y is dying, first case, and here this is dying. So what about this? This will die or not? If this particular is dying, whether this will die or not? Okay, let's leave it. Now tell me, this is about the recessive lethal we are talking about or the dominant lethal in the first case? Only when the homozygous is dying. Okay. So this will die, small y will die. It means that it is recessive lethal. Why it is recessive lethal? It's not because of the reason that the small y is dying. Why it is recessive lethal? It is because of the reason that only homozygous is dying. Remember and know the difference. Only homozygous is dying. So what if both homozygous and heterozygous dies? Capital Y, capital Y, capital Y, and small y, small y. In this, what if this both dies? Is it the correct representation or not? Tell me whether this is the correct representation or not. First thing. Yeah, quick. this part where I've okay, no. So if this dies, if this both the things, both the different combination dies, whether one is heterozygous and one is homozygous. Okay, all of you are saying no. Anyone who is saying yes, I need someone who can say yes. Yes, Manthan is saying yes. How Manthan? Can you just put it in words? It is dominant. Yes, Kusum also saying it is dominant lethal. So students, try to understand it. Don't directly look into the alphabets, whether it is in the capital or small. Try to understand the concepts. The concept when we are talking about the recessive lethal and dominant lethal is, recessive lethal are the one where in all the different combination only recessive dies, only one dies, that is homozygous dies. Only homozygous, if it dies, that means the lethality is recessive in nature. But when both the heterozygous and the homozygous combination, irrespective of whether it is capital Y or small y, if it dies, then it is called as dominant lethal. Now do not scratch your head like what is it, like all concepts is like now destroyed. Don't think like that. Just try to remember like what I have said for now. After that you will definitely understand like what I mean to say. Okay, remember, when we talk about the recessive lethals, it means only homozygous dies. When we talk about the dominant lethals, it means like both heterozygous and the homozygous dies. Okay, fine, good. Okay, so same thing, lethal genes I have explained here. Okay, that's done. And semi till super vital are done. And there are also the other classification of the lethality when it called as codominant lethals, balanced lethal, condition lethal, and gametic lethal. Try to read all of this once. It is very easy to understand, nothing much complex in this. Try to read it. So here in the gametic lethals, the examples is given is it can be the one which I told you about, the meiotic drive or the meiotic drive or segregation distortion, where the one allele will make the another allele not to go for the next generation. See, if some allele is not going for the other generation, what does it mean? Does it mean that it is lethal? Yes, right. So that's what the gametic lethal means. Okay. So theory, next we come to the very important theory, which is the chromosomal theory of inheritance. See, whenever I ask about, like, everyone are familiar with this concept, everyone knows the term. Yes, the chromosomal theory of inheritance. Everyone knows the names also. Like everyone will say that it is Sutton and Boveri, and everyone knows the name of the T.H. Morgan. Everyone knows about these two. But what about the meaning of chromosomal theory of inheritance? Can anyone put it in the words quickly? What is the meaning of chromosomal theory of inheritance? Genes present in the chromosomes. Genes are located in the chromosomes. Okay, good, very good. The behavior, similar behavior of the inheritance of the genes and the chromosomes. Yes, 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 very, very good. So this is what the chromosomal theory of inheritance means. See, although like the name is not explaining what exactly it means, although not clearly, this is what it means. It means that genes which we have studied from the laws of Mendel, what we have studied about the genes, we have studied about the nature of a genes, right? Like how the genes will pass from generation to generation, how it pass. For example, there is a one gene with the two different alleles, so it will separate from each other, form the gametes, and there will be random association from the other parent, and that's how the genes pass from one generation to another generation. That's we have known about the nature. Next, similarly, like how the chromosomes pass from one generation to another generation. Based on the cytological study, we know that there are homologous pairs of chromosomes, and this homologous pairs separate during the cell division and they pass on to the different cells, and that's how the chromosomes pass from generation to generation. This we have known. But the relation between these two was not known until the proposal of this chromosomal theory of inheritance. That's the beauty of this part. This also provides the physical basis of how actually the heredity is following, of how the traits are passing from one generation to another generation. So there are two important names here, two important class of scientists. One is Sutton and Boveri, and one is T.H. Morgan in 1910. Can you tell me the exact contribution of these two scientists, these two categories of scientists, where I have classified into first? together, Sutan and Bovary, and next the T.S. Morgan. So here it is like they proposed this chromosomal theory of inheritance in 1902, 1903, like whatever might be the year. So these are like bit confusing, but remember whatever you know from your books. Okay. Next, the T.H. Morgan. What he has done, he was the first person to give the evidence for this particular theory. So after, remember it as first evidence. It was because of the, it was in the sex linkage concept where this evidence was derived, and it was for the white eye gene of which organism? It was for Drosophila, right? So apart, like after this first evidence, how many evidences we have got till now? Like they are listed, like you can derive any number of evidence, but in the BDC, like we have listed about 10 evidences for the same. And the very next evidence, which is the second evidence, was given by whom? By the student of T.S. Morgan. Who was he? And what was the concept? Remember, second evidence I am talking about the Kelvin bridges. So he gave the non-disjunction, which was actually the exception of, which is also exception of the Mendel first law, based on the non-disjunction theory, where you can see that the chromosomes are not separating while forming the gametes. The two chromosomes are going with the same cell. Okay, they are not separating. That's what the non-disjunction theory explains. If you do not know about the non-disjunction, where disjunction is a fact that the two things will separate. Non-disjunction, where the separation is like failure of separation. You can see the failure of separation where the chromosomes are not separate. That's what the non-disjunction means. Similarly, like you have the evidences of attached X chromosomes. So what is important for your exams? You have to know, like read all of this evidence. If you cannot read it from the BDC book right now, just read it from this PDF, in which like I have mentioned the 10 different evidences. So this evidences will be very much important, because you can expect either assertion reasoning question, or either you can expect the statement wise question from this. Okay. And also remember, like it is LV Morgan. Who was LV Morgan? Morgan's wife, and it was regarding the attached X chromosomes. And also, what is attached X chromosomes? This is also important. Name itself suggests the two X chromosomes are failed to separate, and they will be always together. So this is what the attached X chromosomes means. Next, I'll skip all the evidences for now. Just have to look at it once. Next comes the another important concepts, which is again the most important. Important in the sense that doesn't mean that others are not important. Important in the means like you can definitely expect one question from this part. That's what I mean. So here we have like the gene interaction and the epistasis. What is gene interaction and epistasis and gene interaction? This was also exception for Mendel's first law or the second law. Which one is it? Epistasis or the gene interaction, whatever we say. It is exception for the Mendel's first law or second law? Quick. Yes, it is for the second law. Why? Gene interaction or the epistasis comes into play when we have more than one gene, when we have two or more genes, because we are talking about gene interaction, not the allele interaction. We are talking about the genes interaction, A with B, B with C, and A with C. So when there are more than like two or more than two, we talk about this interaction. Epistasis is the interaction where epistasis generally means the masking of one gene by another. That's it. Okay, the literal meaning of this epistasis. Based on this gene interaction, there are certain defined set of phenomena, interaction that have been listed out. So one is dominant epistasis. So what is the other name of dominant epistasis? If you know, don't tell it from here. Just know it from here, because in the exam they won't be asking you the one name. They can ask you the alternative name for this. So what about dominant epistasis? What is the other name of it? How many? Yes, quick. So what is the other name of this epistasis? Dominant. Yes, it is masking gene action. Recessive, like I'm seeing that a lot of you are not able to answer this question. So please try to remember both the like alternative names on and also the common name that you know. Right? The recessive epistasis is a supplementary gene action, and the duplicative genes with the cumulative effect, it is the polymeric gene action. And next, the complementary, and you know about inhibition, and you know about duplicate. So this has been repeated here, the supplementary, so you can ignore it. Okay, so coming to the ratios, you have to remember the ratio for the masking gene action. It is like 12:3:1. For the supplementary, it is 9:3:4, and for polymeric it is 9:6:1, and for the complementary it is 9:7, and same thing for supplementary, and 13:3 it is inhibitory, and duplicative it is duplicative it is 13, 15 is to 1. So one thing is about the ratio that you have to remember. See, even I have, this is very easy, like from I am doing it from last 3 to 4 years, I can say it easily, and you can also do it from 1 or 2 reading. So you know the names, you know the ratios, plus also make sure to know the test cross ratios here. Okay, and along with the test cross ratio, examples can be important, at least try to cover from the BDC. Whatever examples that have been listed in the BDC, I also try to list it here. But other than that, if you get any other examples from the books you read, from the notes you have, please try to remember all of that, because there is some expectation that it can be asked in the coming days. Okay, the environmental effect of development of character. So now, if I ask you this in a very simple words about the phenotype, phenotype is something which we are able to see, which we can see. Okay, now tell me about my color. I'm a black, brown, or white. I'll give the 3 different options for you people. Just select, choose one. Black, brown, and white. Pick brown. Okay, it's brown. So how am I brown? It is because of what? Genes. It's because of the genes I'm brown. But tell me, like, is it because of only genes, or is there also effect of something else? Yeah, it's melanin. Like, let's take a one thing, general thing. It is because of genes. Okay, will not go in depth. Genes plus environment. That's what I'm trying to say. Environment. See, if I'm born in Delhi, like if I'm been in Delhi for long time, so that also makes the change, certain change in the color, not all to completely. But if I am born in Delhi, then my skin tone color will be mostly like just like albino, like where the melanin production will be not much higher, where the skin color will be very much light. So what I mean to say is, even the genes will majorly affect the phenotype, which we can write it as genotype. But plus genotype, there is also some contribution from the environment too. So this is the equation. So this was first time proved by who? That environmental effect on the traits. By what experiment? The experiment is also very much common and it's very famous. What was it? I have already given it here. Know the name? Know the spelling? It's Johnson in 1911, in which he has given us some very famous theory which is called as pure line theory. This theory was given in the crop of Rajma, which is Phaselis vulgaris, and the variety named the Princess variety. Just know these names, these are very much important. Okay, and what is the experiment, how he came into the like conclusion, these things you don't go for it now, at least for now. If you know it already, it's well and good and it's good. But for now, if you are not aware of it, then do not go for it. Plus, if you have already known, if you have already gone through it, then revise it. Revision is not a problem. But if you are seeing it for the first time, do not worry about it. Okay. Next, environment. Environment of or two kinds. So I have listed out the environment, like 2 different kinds. One can be like external, and other one is internal. Okay. Okay, fine. I missed the year. Okay, so this is 1903. Okay, make a correction on this. I'm very poor at years. Okay, so next thing is about the environment. Environment can be of 2 different kinds, like external and the internal. And when it comes to the external, again it can be divided into abiotic factors and the biotic factors. If you talk about the abiotic factors, it is about the temperature, light, nutrients, moisture, water, and oxygen. So I have tried to compile it only in the 2 slides. Just go through it, because one example I'll tell you. Like in case of temperature, how it can affect. In case of Himalayan rabbits, CHCH, it is recessive allele, provides the fur color at the extremities. So this is providing the fur color at the extremities of the body because of the melanin production, and this melanin production is actually temperature sensitive. So what does it mean? Only in the colder regions where the temperature is not too much, it's very low or it's below the optimum, only there this particular trait is expressed. If trait is expressed, how melanin production takes place. If the temperature is very high, there will be no melanin production. So instead of chinchilla, there will be albino. So that's how the temperature is affecting the trait. Okay. Same thing. Next, we'll talk about the biotic. In the biotic, maternal relation is the one important factor that will affect a character. And in this, one important examples everyone should know, it is like erythroblastosis fetalis. So, how many of you are aware of it? Tell me yes or no. If you know about this. This erythroblastosis fetalis. This is a condition or the disorder when it occurs. At what condition this disorder occurs? This disorder occurs when the Rh positive father is married to Rh negative mother, and this couple have a baby who has Rh positive baby. So when there is a baby of Rh positive from the female of Rh negative, this problem occurs. So what is the exact problem? So problem is, again, the problem is also very simple. So here you can see that this is a fetus, and plus indicates the Rh positive. Again, what is this positive and what is this negative? Rh negative and Rh positive, what does it mean? It simply means that it is talking about the antigen. If the antigen is present, it is called as Rh positive. If antigen is absent, it is called as Rh negative. So here the mother is Rh negative, that is like minus here, and the son, like the baby is like positive. So during the delivery, the first delivery, if you talk about, the some antigen can cross the placenta and reach the mother body. Okay, so when someone is exposed to the antigens that they do not have inherently, so what will be the action that is taken from the body? So what will be the action that is taken from the body? The body will prepares the antibodies, right, against that antigen. If you remember, prepares the antibodies for the Rh positive antigen, right? So because of this, you can see the green ones. These are the antigens, sorry, antibodies which are prepared for that antigen which she was exposed previously. So that will actually create a problem in the second delivery. The second delivery also, like if the baby is again Rh positive, then this antibody can easily cross the placenta and they make the baby anemic, okay, where they will destroy all the RBCs. So this is something which should not occur. So this is the condition which is one of the example for the biotic factors. How it is rectified, I want you to answer in the comment section. So how this can be rectified, and this is a very usual condition. This is not a rare condition. So how this is rectified? Okay. The next one we have also the examples for parasites, symbionts, like population density. So this is also there, like you just have a one reading of it. Next, talking about the internal environment. Internal environment, age. Age will also have some effect on the expression of the traits. I'll tell you about one effect. So can you see the baldness here in my head? I can see the patches here, right? This is the baldness, but this is obviously because of the reason that my ancestors might be bald, so that's why I inherited my character. And for me, like in the real sense, my father is bald at now. But this trait, when it is expressing, I won't be bothered now, because this baldness is a trait which usually expresses after like 30 plus, 40 plus, or 50 plus ages, 50 plus years to be practical, right? So this is how it is. Now I'm not completely bald, okay? So this is how it is. So this is the example of the age. The age actually specifically have some role where some traits will be expressed in the early age and some traits will be expressed in the later age. Another very common example, it is about what? Diabetes. You know, diabetes, the traits might inherit from the like ancestors. Even my father has a diabetes, but doesn't mean that I will be having the diabetes from the day one, right? In the later stages, based on the diet we keep, the environmental effect on a trait, how the phenotype is shaped. So based on what kind of diet I make, what is my lifestyle, based on that, then when I will be getting the diabetes, that varies, right? So this is how the age, one examples, like there are also other examples for it. The next, about the sex. Sex, this is again very much important. So here you have two different types here. Can you name them? Sex limited traits and the sex influenced traits. So this is also one of the mostly confused part for you people, for many. If it is not for you, then it's good. So this is what the thing is. So I'll talk about the sex influenced and the sex limited traits. So what exactly they are? So baldness, remember the baldness which I have talked about, it is the example of the sex influenced traits. See, what is sex influenced? I'll tell you. See, tell me, baldness is only for the men, or it is also seen in the women? Okay, at least someone is laughing because I'm very bad at cracking the jokes. Boys, okay. Women also, fine. Baldness is specific to men or women? Yes, I want answers from everyone. I want answers from all 94 people. Mainly men. Mainly men. Okay. Recessive in female. Yes. I am talking about the expression, whether it is present in both males and females or not. Yes, it is present in both. So that's why it is under sex influence. See, when traits are under sex influence, that means the traits are expressed both in males and females, but the expressivity or the extent of expression varies. Why it varies? Because of the dominance relationship, where in the men the baldness is actually dominant. So if I say the baldness is dominant, that means simply it is getting expressed in both the genotypes, which is homozygous and heterozygous. When I am telling about that it is recessive, that means it only expressed in case of the homozygous. If you can relate, this is what I told you in the dominant lethals and the recessive lethals. That's what you have to remember when we talk about the sex influence traits. That means it can express and it will express in both male and female, but the dominance relationship is different, where in the male the capital B is be dominant and in the female capital B is recessive. That's why in the male we get like lot of, to the greater extent we see the baldness, and the baldness limited to the females, the extent of expression is very low. Okay, it is there, but extent of expression is very low in case of females. That's about the sex influence. You have to also remember about all the other examples that you have seen in the BD Singh book. Okay, so that's are very important. Next, talking about the sex limited, as a term itself suggests, it is limited to one sex. Limited to one sex or limited to male? What is sex limited traits? Is it limited to male or it is limited to one sex? Both. Okay, limited to one sex. It's not limited to male or limited to female, right? It is limited to one sex. It can be either male or it can be either female. So here one good example is like the examples are milk production and the eggs production. As you know, it is limited to the females. These traits. Okay, that's how that's what the difference between the sex limited and sex influence traits. I hope like you won't be forgetting after watching this video. So in the exam, if it is asked, you have first thing what you have to do, you not remember the concept, you have to remember me and what I've explained here. Okay. Next thing, like there are also other things like substrate, tissue specific expressions, and then comes a one important or different term called as phenocopy. What exactly is the phenocopy? I'll explain this. I need some blanks place. Okay. Phenocopy. What exactly it is? For example, let's take a gene which is A gene, which is having two different alleles, capital A and small a. And if I make out all the combinations, it is capital A, small a, small a, small a, and small a, small a. And if it is red, if it is also red, and if it is white, okay. Phenocopy is such that because of the environment, the expression will changes. How the expression changes? The certain environment changes the expression of a allele of a gene to the extent that the resulting phenotype will be exactly same as from the other allele. So if I have to talk in terms of example, if I've showed here the small a should be white, but because of the change in the environment, because of the effect of the environment, what if the other one which has to be red changes to white? These two can be called as the phenocopies of each other, because as a genotype they are not same, but as a phenotype they are same, that is like copies of each other. So that's what it means. And one thing to remember about phenocopy, so is it for single gene we are talking about, or it is between the genes? Phenocopy effect. Altered phenotype due to the different environment. Okay, one simple definitions. Not I'm asking about generation, whether it is for a single gene I'm talking about or between the genes. This example I have given you here. Is it like capital A and small b, that the whatever phenotype it is producing, these two are this combination is producing? For example, if it is producing red, so the other gene is producing capital B, small b, and the small b. If it is producing some other thing, like if it is producing some other color, let's take brown. So if this is altered to red, so this is what we call it as phenocopy. These two will be called as phenocopies to each other, or is it for limited to the one gene which I am talking about? Yes, I am talking about the single gene. The phenomena of the phenomena of phenocopy, it is explained for the different alleles of a single gene. Do not get confused. Okay, it is not between the genes. It is for a single genes for what it is explained. Phenocopy. Okay, so this is done. And next thing, we come across a term called as penetrance and the expressivity. Expressivity, I told you in the previous slide about the baldness, the extent of expression will be different. So now in the simple words, what is this penetrance? Penetrance simply tells us whether the expression, if I talk about the expression, if whether the expression is there or not. Whether the expression is there or not. It is just like yes or no. If I take the 100 individuals, if there is certain trait, whether trait is present in all 100 or not. If it is present in all 100, first case, what it is called as? Okay, so let's take the example of 100 individuals. So if I am just following certain trait. Now out of this 100 individuals, the first case, if all 100 individuals have the trait I am talking about, let's take it as color, red color. If all 100 individuals get that red color from the previous generation, so what should I call it as? Can I call it as complete penetrance? Yes. Can I call it as complete penetrance here? Okay. So if in case, the second case, 90 individuals get that red color and the 10 individuals do not get that red color, so can I call this as incomplete in penetrance? Whether it is yes or no, right? So can I call it as incomplete penetrance? Okay, so this is about the penetrance, where we are talking about the expression of a trait, whether it is expressed in the individuals or not. That's it. But when it's come to the expressivity, the same thing, the same example. If in case that out of 100 individuals, all 100 individuals are showing same extent of expression, then it is called as what? Uniform expressivity. Okay, but if in the individuals some certain category, like let's make it as like some grouping based on the expression, out of 100, first 45 are like 45 of the individuals from the 100 are showing one kind of expression, and rest 20 are showing the expression is there, the red color is there, but the extent is different. The something is providing, some are providing the dark red, some are providing the light red, some are providing the medium red. So let's take it as dark red here. Okay, DR, and next it is medium red, and the rest are providing the light red. So if this is the case, can I name it as like variable expressivity? Yes. So now when the traits are having the variable expressivity, tell me in terms of the penetrance, this example have the complete penetrance or not? This particular example which I have divided into 3 different parts, whether they have the complete penetrance or not. Let me give the number. If it is 45, 20, which is like 65, rest 35, all 100. All 100 I have listed it here. Yes, it is having the complete penetrance, because all of it, irrespective of what, how much color it is showing, but it is showing the color. Showing and not showing will talk about the penetrance. How much it is showing, that will talk about, that is spoken by the expressivity. I hope like this is clear to everyone. Okay. Now we have done some, like this has been like human character study. Since the genotype, the effect of environment cannot be studied, only it can be done in the twins, since it is from the same mother or same fertilization. So this can be only study that can be done regarding the humans in case of with respect to the environment, it is about the twins. We can do it only in the twins, not possible in the other cases. So here, even in the twins, like there are two different types. You have to know about this. These two different types are very much important. You have to know about it. So can you tell me like what is identical twins and what is the difference between the fraternal twins? See, identical twins, as you can see, the zygote is single and it will always have the same sex, and it is from the single sperm and a single egg. But you can ask me, so how come like cell and single sperm and single egg can produce the two babies? It is from the cleavage at the later stages once the fertilization is occurred. So here it is called as identical genotype. It will have exactly identical genotype when it comes to the identical twins. But when we talk about the fraternal twins, it is because of the two different zygotes that you can see here, and also like it can have the different sex. So tell me, like it will always have a different sex, or it can be same or different? Both are possible, or is it the case that always like there will be different sex? Yes, not always. You have to remember this. Okay, it can have the different sex, but not always. And it is from the two different zygotes and from the two different sperms. Remember, it is from again two different sperms fertilizing the two different eggs. That's what the fraternal twins means. So up next, for studying the environmental effect, we take two important terms into consideration. That is concordance and discordance. Okay, I won't be explaining this in detail in this marathon. So if you are interested, then you can just know a bit of information regarding this. So there are quite few chances that this can be asked in the exams. So if you are already thorough with this all these things, then you can give like another extra like 20, 15, 20, 25 minutes and read this concordance and the discordance. That's it. Okay. So for now, for exam, like at least you have to know this. This is very much important. Okay. Inheritance of quantitative traits. So whatever we studied till now, whatever the Mendel explained in his studies, or the 7 characters which was studied by the Mendel, it was related to the qualitative traits. Now we approach to a term called as quantitative. So what is it? It's very simple to understand, nothing much complex. It is the traits we are talking, it is by the multiple alleles or multiple genes here. So here in this case, this is because of the presence of the multiple genes, where a single trait or a single character, it is possible because of having the multiple genes in the back. So that is what the quantitative trait means. And this study of this quantitative traits, it is called as biometrical genetics or the quantitative genetics. And the first serious attempt for that was made with respect to this particular term was the Francis Galton, and later it was Yule in 1906 and Johnson when he gave the pure line theory. And next, most importantly, it was by Nielsen LA in 1908 and 1909. He presented the hypothesis, multiple factor hypothesis. But again, you will tell, sir, you will provide the multiple factor hypothesis. Now I am saying that Nielsen LA presented the multiple factor hypothesis. So which one is true? Both are true. The idea was given here, later it was presented by Nielsen LA. Okay, clear. And next, like EM East and Emerson, they made an extensive, like they collected the extensive data and made some good analysis of this quantitative traits in the later years. And it was in specific some crops, it is tobacco and maize, and Nielsen LA, it was in the wheat and oats. Try to remember the crops also, so that if they are trying to confuse you in the exam, you won't be confused at all. Okay, this is about the multiple traits. So what does the multiple factor hypothesis tells us? See, if we consider this as a qualitative trait, the ratio would be like 3:1 for one gene, 15:1 for two genes, and 36:1 for three genes. Okay, if we are considering like different traits and different traits and different genes. But if we are considering the different genes but the same trait, so same trait, but if we are trying to differentiate the genes, if it is for one gene, it is 3:1. If it is for the two genes, it is 15:1. For three genes, it is 63:1. But what they observed, when they observed clearly, even this 15:1 ratio can be again divided into 6 into 1:4:6:4:1. How? Based on the intensity of color. As you can clearly see, the intensity of the color is decreasing, and lastly we have one category which is white and one category which is dark red. That's why it is 1:1. This is what the multiple factor hypothesis says, where the intensity of color differs and the ratio will not be exactly like 15:1. It would be like 1:4:6:4:1 depending on number of genes if it is for the two genes. Okay, so this was explained in the multiple factor hypothesis. I am not going to go in depth, but two important terms I'll let you know where you might be confused, either going with the BK Prasad BDC book or with the like mock test that you attend. So one is the positive allele, the other one is negative allele. So what are these actually? The positive allele is something which is contributing for a character. That is called as positive allele. What is negative allele? The other allele which doesn't produce any color. If we are talking in terms of color, so the allele which do not have any effect on the trait, that is called as negative allele. So how we are going to get the dark red? Dark red, it is because of the single gene. How we are going to produce it? Like how the dark red is produced? What is there any difference that having 2R and 1R, like having 4R, 3R, 2R and 1R? Is there any difference here? The trait, the amount of trait which is produced, is there any difference between these four? Having 4 genes, capital R, and 3 genes, 2 genes and the 1 gene. So the difference would be in terms of expression. How it is explained? It is explained that each of this positive allele will have the cumulative, that is additive effect. So that's why since 4R is producing the dark red, if you observe, and this part which has 3, see I'll list it down here. So this has 4R, this has 3R, this has 2, this has again 2, 2, 2, 1, 1 and 0. Now see what are the, whether these are correlating or not. One which has 4, it is producing dark red. One which is having 3, it is both are producing medium red. One which is 3, like sorry 2, it is producing medium red. One which is 1, it is producing the light red. And one does not have the positive allele. It is not giving the white color. It is giving no color. Okay. So this is how this mechanism can be explained, that how the quantitative traits actually govern the single quantitative genes govern the single trait, or the multiple genes govern the single trait. Okay. Next comes the QTLs. QTLs are nothing but the location on a gene where the genes for a certain trait is concentrated. Where the genes for a certain genes is concentrated, that particular region we call it as quantitative trait locus. That's it. And it is from the Latin word the place. It means the locus means the place. Okay. And here the what type of curve we see for a quantitative trait, it is always the bell shaped. Remember the bell shaped. Okay, there will be a continuous variation for the trait. Where it, if we talk about the size, if the even though like average size of a seed, if I tell it as 450, doesn't mean that if there are 100 seeds that are produced, all of them will be having the 450. It can range from 300 to 500 or to any number. There will be a range. Okay, this range can be got into a bell-shaped curve. Okay, this is about the QTL, and I have also introduced one term here, molecular markers. So molecular markers are nothing but something that can be noticed or that can be easily detectable in case of the enzyme, in case of the chromosomes, so that we will know the exact location of a gene. That's in a simple words. But in our classes, like other mentors have clearly explained about these markers in the plant breeding class. So if you do not know anything about markers, at least make sure to know about the dominant and co-dominant marker and why they are called so and what are the examples. This can be asked in the exam. Okay. Linkage. Talking about the linkage. So linkage, I told you in the while explaining the second law of Mendel. So while explaining the second law of Mendel, I told you this is an exception. Why it is an exception? What does the linkage means? Linkage means association between the two genes. If this is the chromosomes which I am talking about, this is a chromosome which I am talking about. So here there are two genes which are located to each other. So if these genes are located very close to each other, then in the progeny there won't be recombinants. To put it in an example, let's assume that we have in the parents there is round and yellow that is crossed with wrinkled and white. Okay. Now in the progeny, if I tell you that there are 100 individuals in the progeny, but what are the phenotypes of that individual? That they are round yellow and wrinkled white. So now observing this, what conclusion that you can come up with? What conclusion you can make? Yes, fast. So based on this observation that from the parents to the offsprings, complete linkage. How come you said it as complete linkage here? By considering what factor? Very simple, right? See here, can we call it as parental type? And this is also parental type, because why it is called as parental? Simple, it is present in the parents and in the progeny. What we are observing? Are we observing only the parental type? Are we observing any recombinants here? No. Why we are not observing the recombinants? That if you want to understand that, then we have to go for the chromosomal structure here. Okay, let's assume this is P1 and this is parent 2. Let's assume we are taking the one chromosome here, and let's assume that genes for the both the traits are present in the same chromosome. Let's assume it as capital A, small a, and small b, and this is small a, small b, and small b, small b. So here, if we want to get the recombinants, then there should be a crossing over between these two, where it will give us capital A, small b, capital A, capital B, capital A, small b, capital A, sorry, small a, capital B, and lastly it is small a, small e, where last two are the parental, are the recombinants. And if this crosses with the other one, which is obviously that only one combination, then there is a chances that we get the recombinants, right? So if we are not getting it, that means the crossing over is not taking place. The crossing over, if it is not taking place, that means the two genes are tightly linked with each other. That means this is the phenomena of linkage, where the offsprings are like not having recombination. So again, in this 100 individual, if I take your second case where I tell you that the 90 are parental type, rest 2 I have round with white and the wrinkled with yellow. So these are totally of 10. This can be this 5 and this is 5. So now tell me like whether there is a linkage or not. Yes or no? First thing, whether there is linkage or not. So what do we call that linkage as? So what can we call this linkage as? Can we call it as incomplete linkage? Ignore my handwriting. It's not good, I know. But still, I hope like everyone are able to understand the concepts which I am explaining, right? Should I write a bit slow and should I write it very nicely? Is it required? I don't want to waste your time. That's why. Okay, fine. So this is about the linkage in simple words. See, in all my classes, although like many things are present in the screen or many things are present in the slides, I won't simply read it and underline the things and explain you. Like I do it in my way, like where I explain you the terms in a very simple words. I don't give it in a detail insights about everything because it's not required. If I just give you a keyword and try to explain the keywords, so the rest of the things you can read it by your own. Okay, fine. Next thing, linkage. I told you it is about the genes which tend to stay in, together, and the Bateson and Punnett. You have to know about these names who are the one to first propose this linkage concept. And again, when we talk about the linkage, there are again two different terms that are needed to be known. So what is it? It is about the coupling phase and the repulsion phase, and these two are the phenomena of the linkage only. So what is coupling and what is repulsion? So if I have two genes, if talking about the coupling phase, so what does this coupling phase means? It means the two genes, the two dominant alleles will be at one chromosome, and the other one will be at other two which are recessive will be at other chromosomes. And if other chromosome is not other, like homologous chromosome, and the repulsion, it is like in the one of the homologous chromosome it will be capital A small b, and the other will have the small a capital B. So these are the two different phenomena of the two different terms under the linkage concepts, which is called as coupling phase and the repulsion phase. So this is very much important to understand. I'll tell you why in the later slides. Next, so similarly, apart from the linkage, I introduced a term called as crossing over. You know about the crossing over. Crossing over, it is about the phenomena, or it is an event which results in a phenomena called as recombination. Okay, so this is an event which is occurring, because of which there is a phenomena or the result that we are seeing, that is recombination. Now what is this crossing over? Which is the exchange of strictly homologous. The term is very important, it's homologous. So how to calculate the crossing over? The frequency of the crossing over is nothing but the recombination frequency. It can be calculated by the number of recombinants in the test cross divided by total number of progeny. If you are for the PhD, then these questions will be also asked, but for you, at least know the formula. Like for PhD, like 2-point test cross, 3-point test cross, all of these are important. Those concepts have been just introduced to you. Just know the formulas, that will be sufficient. Next, what are the factors affecting? If I say that the two genes are present on the same chromosomes, whether the recombination can happen or not. So there are certain factors which will actually influence the recombinations. So that can be the distance between, this is obviously true, that we have already known. If far the distance between the genes, the easily can they undergo crossing over or the recombination. So that's why the distance will matter, the sex will also matter, the age of the female, the temperature. So try to remember all of these things, read it once. So in case of statement wise questions, if it is asked, or the assertion reasoning, if it is asked, you should not skip it. Okay. Cytological basis of crossing over. So what I just explain that term for you. What is this cytological basis of crossing over? I told you in the definition of crossing over that there is a strict exchange of homologous segments. But did we visualize it? Do we know it visually that, okay, if there is a chromosome, homologous chromosomes, if there is a crossing over here, then this parts, these physical segments have actually undergone crossing over. So have you visualized it anywhere? How do we know that there is strict exchange? Yes, we know from the traits that recombination is happening, but recombination can be by any different phenomena also. How come it is, we are saying that it is because of the strict exchange of the segments? That is what the cytological basis of the crossing over explains. So who explained this cytological basis? It was by the Stern in case of Drosophila, who explained it for the first time in the example of carnation and bar. Okay, that during the crossing over there is a physical exchange of the gametes. Okay, that you have to remember. And next, what are the other, if I ask you about the other examples, I already shown you. Okay, the second example is Crichton and the McClintock. It was explained in terms of, in case of maize. Okay, so first, and it is 2. In case of maize, the same, the cytological basis that there is a physical exchange of the chromosomal segments in the crossing over is explained in this 2 different experiments. See, like irrespective of whether you know the experiment or you understand the experiment is secondary. You know it or not, that doesn't matter right now, but at least you have to know the examples. If they are asking about the Stern provided the cytological basis for the crossing over, you should be knowing, yes, he provided it. So you should be able to link it for now. If you already know it, just try to recall and revise it from the PDF. Do not skip it. Again, I'm repeating it. If you already done it, don't skip it. Revise it. Revision will not take much time. But if you have not done it before, you have not understand it before anytime, anyhow, like all the students of the master course will be able to understand it because I explained it clearly. But for others, if you have not understood it for right now, you can skip it and try to remember the example that cytological basis of the crossing over is given by the Stern experiment and also with the McClintock and the Crichton experiment in the maize. That's it. Next one. These 2 important questions need to be answered. First thing, when does the crossing over takes place? When does the crossing over takes place? It is at the 4-strand stage or the 2-strand stage. See, if you know the chromosomes, although it is homologous, but because of the S phase duplication, it will be divided into 4 different strands. So now the question first is whether it is in the 4-strand stage or the 2-strand stage. Why it is important? Because if it is in just 2-strand stage, if the crossing over is taking place, and in the other case if it is in the 4-strand stage, the combination we get here in the both the cases are different. It is not same. So that's why it is important to know if the combinations are different, then result will be different. That's why it is important to know whether it is in the 4th strand or 2 strands. Tell me whether it is in the 4th strand stage or 2 strands. Yes, it is in the 4th strand stage. Remember it. And it was answer was given in the Neurospora ascospore column. So based on the experiment that was conducted in this organism, this was proved. Next thing, the relationship between the chiasma and the crossing over. So what is chiasma? If you know about the chiasma, if this is the chromosomes, whenever the crossing over is taking place, after the crossing over has been taken place, there will be certain X-like configuration that is present in the chromosome. Now I know, that's why I am telling that it is after crossing over this configuration taking place. But initially when these concepts were introduced, there was not a clear-cut evidence whether the chiasma occurs first, or what I mean to say is whether the crossing over is because of formation of X or the X is formed because of crossing over. X is formed because of crossing over, or chiasma is formed first and then crossing over has happened. So this was the question, and later it was resolved. Before that, this is explained between the 2 different theories, which is like classical theory and the chiasma theory. According to the classical theory, what it explains, it tells us that chiasma and the crossovers are followed by the breakage. That means the chiasma occurs first and then there will be a breakage and the reunion. And from the chiasmata theory, it says that chiasma is the consequence of crossing over. That means the crossing over occurs first and then the chiasma occurs, which is actually true. Now, based on this, later there are other things which came into existence. Now, I told you about the cytological basis of the crossing over. What about the molecular basis? Cytological basis is in terms of the chromosomes, whether the physical segments are exchanged or not. But what about the molecular? Molecular means in terms of ATGC, in terms of actual nitrogen basis, where there is an exchange or not. This is the molecular basis. So this is explained by the double strand break model. Now, so this molecular basis, as I told you, it is explained by the double strand break model. And even in the break model, there were 2 important models. One was regarding the it was like breakage and reunion theory, the theory which try to explain the molecular mechanisms. What I mean to say, like the physical exchange of segments which is taking place is explained about by the breakage and reunion theory and the copy choice theory. Okay, and this theory, what does it says? Breakage and reunion simply says that if this is the chromosome, if there is a crossing over taking place, there will be a breakage and there will be exchange of segments. That's what the breakage and reunion theory explains. But the copy choice theory explains that there is no breakage, there is no reunion. The new segments which are formed here, they will just copy from the opposite segments. They will have a copy from the opposite segments, and that's how the crossing over is actually taking place. So which is not true, and the breakage and reunion concept is actually true. Okay. Now, based on this, there were hybrid DNA models which tried to explain the molecular mechanism. Molecular mechanism in the sense in terms of ATGC, like how it is actually spread. That was tried to explain by forming the DNA models. And the 2 important models that you have to remember is Whitehouse model and the Robin Holiday model, which was in 1963 and in the 1964. And out of this 2 model, this is mostly used because of its simplicity, attractivity, and the mostly accepted one. Okay, this is well known among these 2, and there are also several other models, but among this, so this is mostly accepted and this is mostly used. This hybrid DNA model explains the breakage and crossing over into 2 different types. So one is the single strand model, where they explain that there is a single strand break, and because of the single strand break, the crossing over has occurred. And next we have a double strand model. So this was from the single strand model, Holiday model, where you can see there is a single strand breakage on the both the chromosomes. So you can take it as a chromosome here for a better understanding, and then there will be a junction that is formed, and then there is a ligation, and that's how the recombinants are being formed here. So this is from the single strand break. But tell me which is proved now. How the crossing over taking place, how the crossing over is taking place, whether it is based on the single strand model or the double strand model. Whether the crossing over is baked because of this is proved based on the single strand model or it is currently with the double strand model. Yes, it is with the double strand model. Difference between the double strand and single strand. Again, I'll clarify this. Single strand crossing over doesn't mean that the single strand break doesn't mean that there will be break only in the one chromosome. See, you can see the break in both the chromosomes. Breaks are there, but not on a double strand. Only one of the two strands is breaking here, and only one of the two strands are breaking in the second one. That's why it is called a single strand. So what does a double strand means? Double strand means the one break is formed in the entire double strand of a DNA. That's what the double strand break means. Based on this model, so we came into conclusion that how the recombinants have been produced. All of these are the recombinants. Okay, these are the possibility. And explaining this in details, I won't be doing it. But if you have read it, again, you can go through it. Okay. Next, the linkage maps and the linkage groups. The linkage map, what is this called? This is also called as genetic map. And why it is like linkage map? Because the what is map? Map is nothing but ordering the genes on a chromosomes, knowing the exact location. And how this is derived? This is derived based on the recombination frequency, based on like what is the percent recombination between the genes. That's why it is called as linkage map. And to construct it, two important information is required. One is you have to know about the recombination frequency, and second, if there are more than two genes, then you have to know about the order of genes. See how to find out the order, how to find out the recombination. I have clearly explained it in the videos, and also like you can find it in the BDSing, nothing hard to read. So if you know it, it's well and good. If you have not known it, just go through it once. This is important, like to calculate the recombination frequency. Okay. And the formula, remember the formula, if it is for the two genes, which is called as 2-point cross, test cross, the recombination frequency is like total number of recombinants divided by 100. But if it is for 3, it is total number of SCO, single crossovers, plus number of double crossovers divided by total number of progeny. So what I mean to say is, if I have 3 genes, if I have to find the distance between A and B, then the single crossover between the A and B is needed, and double crossover here, the double crossover between these two is needed divided by total number of progeny. This will give us the recombination frequency between the A and B. And similarly, I can do it for B and C also. Okay. And here there is a one new term which is introduced. It is called as interference. So what is this interference? The interference, for example, take it logically. There are there are 3 genes which are like almost near to each other. So now if there is any crossing over in this region, will it have any effect whether the crossing over will happen in the adjacent region or not? Whether will it have any effect or not? It will resist the formation of crossing over at a nearby site, or it will be always independent? What do you think? Just a logical question, where like if I have something and there is some crossing over that is taking place and near to this site, whether it can easily form another crossing over, or there will be some resistance for the crossing over to take place. There will be resistance, and this resistance is explained by the term called as interference, where the occurring of one crossover will not allow the nearby region to have the crossing over. So that phenomena is explained by the interference, and the one which is opposite to it is coefficient of coincidence. Okay. Now this too has some very important meaning. Whenever the interference value is 1, so what does it mean? Whenever the coefficient of coincidence value is 1, what does it mean? So those things are important. Before that, how to calculate it? How to calculate the interference and how to calculate the coefficient of coincidence? So coefficient of coincidence, it's very simple. It is just by observed frequency of double crossover divided by expected. What was expected? If you just do it, you'll get the coefficient of coincidence. And how to get the interference? Interference is also very simple. It is just 1 minus coefficient of coincidence. Okay, that you have to remember. And the interference concept was first given by the Muller in case of 1916. So here, as I told you, if the coefficient of coincidence is 1, that means the two crossing overs that are happening here, they are independent of each other. Okay, that is what it means if it is 1. But if it is 0, that means there is a complete interference where occurring of one crossing over will not allow the other crossing over to take place, where it will lead to the no double crossover. There will be single crossover, but there won't be no double crossover in this case. Okay, this is what the 0 and 1 means. If the coefficient of coincidence is between 0 to 1, then what it means, there is a partial interference where the observed double crossover will not be equal to expected double crossover. There will be some double crossover that are there, but it won't be equal. Okay, and if the coefficient of coincidence is 0, then what will be the interference? What will be the interference here? Yes, quick. If I say that coefficient of coincidence is 0, then what will be the interference? It will be 1. So if even if in the exam, if they ask like if the interference is 1, what will be the effect? Effect is simple. There will be no double crossovers. If it is 0 for coefficient of coincidence, same thing. There will be no double crossovers. And here, if I say that coefficient of coincidence is 1, then what will be the interference here? The interference is again here 0. If there is no interference, then will be the observed double crossover will be equal to the expected double crossover. That's it. Okay, fine. I hope this is clear to everyone. And to make some corrections, so since you have understood now, because of the concept of the interference, there is some problem with the recombination frequency that we were calculating to know the distance between the genes. So because of which there are two important functions, the corrections that have been introduced. So this was actually asked in my paper when I gave the exam in 2020. They asked about the features of this aldane in the statement based questions. So remember two important things. In detail, nothing is needed to be remembered, but remember two important things. Aldane function, it won't assume the lack of interference. It won't assume that interference is there or not, nothing else. So the genetic distance here, it is by the log of 1 minus 2 r directly, where r is the recombination frequency. But in the Quantitative mapping, it actually assumes the occurrence of interference. So this is because of this reason, the value of the genetic distance that is derived by the aldane, it will be higher. Since it assumes no lacunas, the distance will be higher. Since it assumes the some interference is there, the value is lower than the above. That's what you have to remember. Only two different things. Once you go for your MSc, try to upgrade this knowledge. So whatever you know from here, try to take it so that most of the things will be very much clear and it will be needed for your PhD exam. But for now, this should be introduced, and you should know there are two important functions to correct the mistake, which is like aldane mapping function and the Quantitative mapping function. Next, so we have come up with the sex determination. So in case of sex determination, what is important? First thing, it was by the Enking who just observed the X bodies which were unknown in the starting, where the morphology of the chromosomes were known, but he observed the unusual morphology, like which was different in morphology. That was first time made by the Enking. Later, Wilson and Stevens in 1905 demonstrated about the presence of the supernumerary chromosomes, which were the sex chromosome. But they were not knowing that it was sex chromosome, but they mentioned it as like supernumerary chromosomes. And later in the Stevens, he gave the Drosophila Y chromosomes was discovered, and the Wilson, he proposed the sex determination. He gave some clarity regarding the involvement of the X chromosome in sex determination. Like for example, like male and female, like how someone is developed as male, how someone is developed as female. So that is linked with certain chromosome, which is X chromosome, was given by the Wilson. And there are three important mechanisms of sex determination here: environmental, chromosomal, and genic. So now in this genic, what you have to remember as soon as you see the genic, it is by the G-link balance theory. I'll tell you what is this genic balance theory. So who gave this genic balance theory, if you remember? Who gave this theory? See, I'll tell you again and again. In case of genetics and plant breeding, the concepts, the respective names of the scientists to whom they are credited to, and the years, and what is the exact principle or what is exact theory they have stated. These things are very much important. You can't skip it. So this was by Bridges. I'll explain in detail about this Bridges in the coming slide. Now I won't consider this environmental, you can read it by your own. So here in case of chromosomal, what are the different things you have? In the female it can be XO crossed with XY male, and female can be XX and crossed with, sorry, this is XX, so this can be XO, and the other combinations are XS female, normal, and XY. Okay, and this can be like XY in the females and XX in the males. So these are the different possibilities. So examples you have to remember: XY in the females and the XX in the males. The example for like ZW and ZZ is example for birds and reptiles. So other than this, we have another classification, which is it? Diploid females and the haploid males. What are the examples for this? Where all males will be haploid and all females will be diploid. Honeybees, right? So these are the different types of sex determinations that you are going to observe. So what is important from here? Knowing the example and knowing the exact mechanisms. Just go through it. Okay. I won't be like explaining anything here. Next, when it comes to the genes, remember these three different genes. When we talk about sex determination, at least we should know like which genes are actually determining the sex. So SRY gene in case of humans and SRY in case of mice or mouse. So it is present in the which chromosome? It is present in the Y chromosome. This is the name of a gene. And this XXL gene, sex lethal gene, it is in the Drosophila. Whether it is present in the autosomes or X chromosomes or Y chromosomes, the XXL gene, where it is present? Yes, quick. Sex lethal gene for the sex determination in the Drosophila, it is present in, yes, it is present in the X. Okay, fine. Okay. So genic balance theory, what does this theory explains us? This theory explains us that the sex of the individual, which is determined as a sex index, it is because of the ratio of number of X chromosomes divided by number of autosomal sets. Do not get confused, this is sets, not just the autosomes. For example, in case of humans, what is the chromosome number? 22 pairs plus 1 pair, right? So here number of X chromosomes in case of human female, how much it is? Is 2. Down, number of autosomal sets, does it mean 22 pairs or does it mean 2? Which one is true? Yes, it is 2. 2 represents the diploid nature, the autosomal sets, whether they are present in the 2 copies or not the 22. Okay, do not get confused. I've seen students getting confused here. So if I just do this, what is the value I'm going to get? 1. That means the sex index of the human female will be 1. So taking into the sex determination theory for the Drosophila, I took a hypothetical example. Don't get confused with the humans. Okay. So here the formula is very simple, that you have to know the number of X chromosomes divided by total number of autosomal sets, which will be equal to 2 generally. So now here you have to know that 1, if the sex index value is 1, it is called, will be normal female, and if it is less than 0.5, it is meta male, sorry, and if it is 0.5, then it is normal male, and if it is more than 0.5 to 1, then it is super, sorry, intersex, and it is 1, it is female. And what if there is a more than 1, then it is super female. So just try to remember the ratio. The ratios are very much important. He gave this ratio that this is based on the number of X chromosomes and divided by total number of autosomal sets. Okay. So mechanism, you can skip it for now. Dosage compensation. This is the another important concepts that you have to know. Dosage compensation. So let's take an example. Human males and human females. Let's only talk about the sex chromosome. So in sex chromosome XY and XX. So now you see about the X. Tell me, X chromosomes will only have a genes for the sex determination, or it also has the genes for the other normal functions. X has only genes for sex determination, or it has other genes for other vital function of the body. Which one is true? First one is true or second one is true? Tell me one or two. If first is true, type 1. If second is true, type 2. It is like plus, okay, both will be true. Okay, fine, that's good. So X will actually have the genes for sex determination, that's true, not in humans, okay, but generally speaking, plus secondary sexual characters will be there, plus it also have the important genes for the other vital function. So if that is the case, then how come that single gene is present in the single chromosome is present in the male and the two chromosomes are present in the male? This, if this has a genes, for example, this has a gene like hey, has some enzymatic function, it produces some enzyme. Same thing, it is in the two copies, there are multiple. So the enzymes here will be like the concentration of the enzymes will be high in the female and the low in the male. But do we see any kind of difference in terms of character expression? The normal, the common traits which are present in both male and the female, is there any difference? We see almost same, right? There is, there is no difference in the males and females, right, for most of the traits. But then how this dosage is compensated? There is a one dosage, there is a two dosage, but at the end whatever traits we have, whatever traits we see, so they are no difference. So this is because of the concept of dosage compensation, where there are three important mechanisms that you have to know. First one is inactivity. So this kind of mechanism is seen in the humans or the mammals, where one of the two chromosomes will be converted as what? It will be converted as the bar body. Okay, so this is in case of inactivation. And next, when it comes to the hyperactivity, one of the two X chromosome will have the higher activity. This is case in the Drosophila. Third, hypoactivity. Two X chromosomes will have like lower expression, lower activity, both of the X chromosomes. And this is observed in case of nematodes. So this is the three important phenomena of dosage compensation, which you have to remember along with the examples. Next, so I'll leave it to here. You can read by yourself. This is not that much important. Okay. Next comes to the another very important part, which is the cytoplasmic inheritance. Okay. Cytoplasmic inheritance, here we have to know, like remember two important things here. I'll go to directly that one. Okay. In case of cytoplasmic inheritance chapter, you have two important terms. One is maternal effect, one is cytoplasmic inheritance. So you have to know about these two clearly, the difference between these two. See, what you know from the cytoplasmic inheritance. Cytoplasmic inheritance says that the trait from the female will be passed on to the generation, and in every generation the female will be inherited, so that phenotype will be seen in every generation. That what we have seen in case of cytoplasmic inheritance. But in the maternal inheritance or the maternal effect, you can see that in the first generation directly you are seeing what is there from the female, right? What is there from the female. But in the later generation, this is not repeated. Or what I mean to say is, in the cytoplasmic inheritance, there is a reciprocal difference. What is reciprocal difference? If the female and the male are altered and crossed, there will be difference in the end phenotype we are getting in the progeny. That reciprocal difference is observed in the female cytoplasmic inheritance. So similarly, in the maternal effect also, same reciprocal difference is seen, where you can see the difference in the progeny that is formed. But this reciprocal difference will last for only one or few generation, and in the rest of the generation both will be same for both the reciprocal crosses. This is case of maternal effect. But when we are talking about the cytoplasmic inheritance, no matter like how many generation you go, always the phenotype will be inherited from the mother only. So this is the one difference. So you have to know the difference, plus also remember the examples for all of this. Okay, so that's most important for you. So here I'll tell you about the few terms. What is plastome? What is chondriome? Plastome is like genes present in the plastid. Chondriome, which are the genes which are present in the mitochondria. Okay, and the plasmon. What is this plasmon? It is like sum total of all the genes that are present in the cytoplasm. And if you know, in case of nucleus, the genes are present in the nucleus, everyone knows it. But other than nucleus, in the cytoplasm, there are only two important organelles in where the genes are present or the DNA is present. It is chloroplast and it is the mitochondria. Okay, remember this. Next, the history, you can read it on your own. I'll just skip it. Next, the non-Mendelian inheritance, which explains about the deviation from the Mendelian inheritance. First thing is reciprocal difference. So this is divided into three different types. One is inheritance of structure and patterns, and the second one is like non-Mendelian inheritance because of the parasites, symbionts, and virus. The third category is actual cytoplasmic inheritance. So now tell me, like this killer trait in paramecium, is it the example of the cytoplasmic inheritance? Yes or no? Is the killer trait in the paramecium? It is the example of cytoplasmic inheritance, true cytoplasmic inheritance, or it is because of the cytoplasmic genes? Is it because of the cytoplasmic genes or the nuclear genes? Which one is true? It is because of the nuclear genes. Okay, remember, do not get confused. And if in the exam, if they're asking about the examples for the cytoplasmic inheritance, do not directly tick this as cytoplasmic. It is not cytoplasmic, but it is under non-Mendelian inheritance. If they are asking about non-Mendelian inheritance example, this is true, but not the cytoplasmic inheritance. Okay, fine. So what are the two examples of cytoplasmic inheritance? Here we have the plastid inheritance. That is the example which is seen in the chlorophyll variegation in the Mirabilis jalapa. This is a true example for the cytoplasmic inheritance where the genes responsible for these traits are actually present in the plastid or the chloroplast. And when it comes to the mitochondria, what is the example? Example, the two important example. One is like petite. The second example is CMS, cytoplasmic male sterility. Remember these two examples. These are the very important examples from the mitochondrial inheritance. Okay, so that's it. So this almost completes the Mendelian genetics. I have left out with one topic which is multiple alleles. I won't be discussing it now. I'm already too much exhausted. So we'll take it in the other session. I hope the whatever we have discussed till now, it is informative for you, all of you, and you have like refreshed all of your knowledge that you have gained and all of the things that you have already learned. Okay, so I guess like admit card has been already released, also released right now. So please download the admit card. So how to download the admit card if you are not aware of it, I'll make a video and upload it in the YouTube very soon. Just follow the process and take the admit card. And after taking the admit card, make sure to read the instruction at least once. At least once read the instruction that is given in the admit card. And I'll tell you like what are important. Take the admit card to your exam. Take 2 photocopies and make sure to have the thumb impression from home only, because at some places you might not find the that ink pad, not at all the centers. So make sure to do it, and rest, that's it. 2 photos, pen, again it depends on the center. Some centers will give it, some centers will not give it. So make sure to carry one pen. Sheet is not required, and even if you carry the water bottle, make sure it is transparent, not the any thermos or any other bottle, because you have to leave it outside. And carrying the bags, laptops, do not carry any like costly items with you. You can carry some books, one book or two books that's needed, and at least reach the center at least 1 hour before the exam. Okay, fine. That's it from my side. I hope like you enjoyed this session. I'm too much exhausted since like it is very first time that I have stayed for 2 hours. If it was on the laptop, I would have done for at least 3 to 4 years. But coming on a smart board, so it's bit hectic. But still, we will try our best to come up with the other sessions too. I'll let you know about the molecular genetics very soon, by tonight or tomorrow morning, whether I'll be able to do it or not. Because there are also other lot of other commitments that students are requesting. Our master course students are requesting, so we have to also look into that. Rest, I hope like whatever I have discussed, that is okay. Thank you for genetics. Yeah. See, students, like if you have if you have read the genetics very thorough, then this session would feels like okay, it's nothing. But the students who are very much like worried about the genetics, they do not know anything. They want like everything to be covered at this very last moment in a very simple words. Then this is very perfect for them. But for all of you, this is almost having like 99 to 100 slides. Okay, so this you can easily read it in 1 hour mostly, maximum in 1 hour. So if you want this PDF, what you have to do, you have to just fill the Google form. Whoever has already filled it, you will be getting it by tonight. The PDF will be uploaded in the same Google Drive link. So if you have not yet filled the Google form, if you are first time to this video, then fill the Google form that is available in the description box. We will make it available after the video is published on the YouTube. Like it will be done once the live stream ends. In the description, go and check the Google form link. So it, by any chance, if you don't get the Google form link, you can just text us on this below displayed WhatsApp number and get the Google link, fill it, and get this PDF, which is like offline printable. Either you can print it or you can read it on your tablet or in the laptop or in the mobile, anywhere which is suitable for you people. Okay, fine. Thank you so much for attending. I hope like this session was informative for all of you, and hopefully like we'll try to come up with the similar kind of sessions in the upcoming days. Thank you so much and wish you all the very best. So the link is available in the pin comment also. If you have, okay, if you have filled the Google form and if you have not got any PDFs, just text us on this below displayed WhatsApp number. Our team will help you with that. Okay, thank you so much for all the love and support that you people are providing us, and in the coming years also we expect the similar kind of support. I understand like your master course is done, you have done your things and you will be going for your masters, but the feedback, but the spread of information that you will provide, that is very much needed and crucial for us to build this platform, to make it to take it to the greater heights, and also to involve all the subjects. So if you're going to MSc, you will be definitely requiring the courses for your PhD too. So if you want us to get the courses for the PhD, the right courses, the right quality, so please make sure to spread this, that about our online master course for 2026 and also for 2027. Get to know about our platform, introduce our platform to the new students, so that we can work for you people and try to provide the best quality content which is actually lacking in the field of agriculture. Thank you so much.